So now we're talking about the seedless vascular plants, which are so named because they don't have seeds and they have vascular tissue, which is very good. I will also be referring to them as pteridophytes throughout this video, probably, maybe, we'll see. So vascular tissue is an innovation by this clade of plants that arose from out of the non-vascular plants, the bryophytes, if you remember. They're called the tracheophytes. Vascular tissue is very good because it allows plants to get off the ground. As you may remember, we talked about how bryophytes did not fight against gravity as they colonized land. In fact, they stayed flattened to the ground and relied on passive diffusion to transport resources. Plants, of course, after a long time of evolving, figured out that in order to get more sunlight, you had to be higher up. So then they grew higher up, but then they had to fight against gravity. Vascular tissue not only provides the structural support that helps them stand against gravity, but also gives them the conducting channels that they need to send fluids and other nutrients from their roots up to the photosynthesizing leaves, and then the products from photosynthesis to the rest of the plant body that needs it. Wood is a very great example of vascular tissue. Wood is mostly vascular tissue. Vascular tissue is also very prominent in this cool vegetable called celery, which you might want to try to eat someday. So sorry, bryophytes, but we're moving on. So these vascular plants, or tracheophytes, first evolved in the late Devonian, but got really big in the Carboniferous. This is an image of maybe what the first vascular plants might have looked like in the Devonian. In the Carboniferous, they almost took on tree-like forms as they spanned huge forests that contributed to the high oxygen in the atmosphere during the Carboniferous and also formed those coal deposits from the Carboniferous. And we'll be talking about the seedless vascular plants in particular. So you may remember how I talked about a Russian nesting doll kind of analogy for this whole plant systematics adventure, where the embryophytes were a group that had a more selective group, which are the vascular plants, the tracheophytes within it. Sorry, clade. These are all clades. The tracheophytes will have a more selective group called the spermatophytes, or seed plants, within it. And we'll be focusing on the plants that are excluded from the seed plants, which are the seedless plants. Just like how in last video we focused on the bryophytes, which are excluded from the tracheophytes. It's important to know that bryophytes and pteridophytes aren't clades by themselves because the vascular plants and seed plants, respectively, emerged out of them, and they are not included in the definition. Oh, and the main two groups of seedless vascular plants are clubmosses and their relatives, and ferns and their relatives, Lycophytes and Monilophytes. So vascular tissue is a pretty cool thing. And it's comprised of two different types of tissue. One is called xylem, that transports water and minerals up from the roots to the leaves of the plant. Xylem is typically more important and more tested on, I believe, and it provides the structural support of vascular tissue in that these cells are lignified. So you may know that plant cells have cell walls. Most of them have what is called a primary cell wall. Xylem and some other structural support tissues, their cells evolve secondary cell walls, which are chock full of an organic polymer called lignin. Lignin is very structurally sound and provides that support that vascular plants need to stand against gravity. This is what some microscopic pictures of xylem look like, comprised of two types of cells. We can talk about them later in plant physiology, tracheids and vessel elements. What is unique about xylem is that these cells are dead. There is no cell within the cell wall. These are essentially hollow tubes in which water and minerals get transported up. Phloem, on the other hand, their cells are not dead, although they are still kind of hollow, and they are also not lignified. Phloem is the conducting tissue that transports sugars and other photosynthetic products from the leaves to the rest of the plants, because not all parts of the plant undergo photosynthesis. Phloem is also kind of known as an information superhighway because things like hormones, chemical messengers, and even plant viruses all are transported via the phloem. So that's vascular tissue in a nutshell. Vascular plants had several other innovations, including apical meristems. Apical means at the tip of something, and meristems are just a group of dividing cells that help a plant grow. So apical meristems are those plant growth centers at the tips of shoots and roots. They also have a sporophyte dominant generation, in contrast to the bryophytes, which had a gametophyte dominant generation. That means for every other plant that is not a bryophyte, the main quote-unquote plant that you see is the sporophyte phase. They have sporophylls, which are modified leaves with sporangia, and more advanced water conservation. This is a sporophyll, for example. More advanced water conservation, as in they mostly all have cuticles. Cuticles are a waxy layer covering the epidermis that help protect the plant lose less water from things like transpiration and evaporation. So I talked a little bit about these apical meristems, but they really contribute to one of the fundamental differences between plants and animals, which I think is really cool. And I'll talk about a little bit here as we discuss plant tissue, because plant tissue really gets big in the vascular plants because they're actually creating specialized plant organs, roots, true stems, and true leaves. Apical meristems, because they continuously grow and elongate the tips of shoots and roots, contribute to the plant's ability called indeterminate growth, which is in high contrast to most animals. See, animals, when they reach a phase, say like sexual maturity, they stop growing and simply maintain their current size. Some animals are so extreme, there are a few worms and like tardigrades that when they reach maturity, they have even a fixed number of cells. They have, say, like 907 cells. Every adult has 907 cells, and each cell is specialized to a very specific use. That is called determinate growth because everything is determined and growth stops at a certain point. Plants exhibit indeterminate growth. Outside of their organs, like leaves, which also exhibit determinate growth, the plant itself can grow indefinitely provided it has enough resources. And that really goes along with this point of specialized plant organs, which are spread throughout the entire plant rather than localized to a single organ. For example, all of our metabolic functions are kind of compressed into a few organs, like our liver. And therefore, if we lost our liver, we die. Plants, on the other hand, have their functions spread across the entire plant body. Leaves perform metabolic function, but the plant has leaves all over the place. So if it loses a couple of leaves, it's not really a big deal. And that's important because, of course, plants are mobile and can't run away. If I was getting my liver eaten, I could probably run away. So that's a very fundamental distinction just between animals and plants in general. Just the entire strategy of which they build themselves and their tissues, which is, I think, is very cool. All plant organs are comprised of three fundamental types of tissue, which are the dermal, vascular, and ground tissues. Dermal tissue is basically the skin of a plant. So the epidermis, the cuticle, which I talked about, any projections of the epidermis, like trichomes, which are like hairs, and also bark in woody plants. Vascular tissue we talked about already. Here's a histology cross-section of vascular tissue where the xylem and phloem are very distinct because the xylem is red. I'd assume that this is a stain for lignified cells. Of course, xylem are lignified, but usually xylem also have bigger diameters than phloem. I don't know. Histology is kind of weird with plants because everybody uses different stains, but in general, you can identify xylem by this. Ground tissue is everything else, everything in between the vascular and dermal tissue, and also performs the actual functions of the plant. So in the leaves, the cells that actually photosynthesize are part of the ground tissue. It is composed of the parenchyma, collenchyma, and sclerenchyma, which we will get into when we reach plant physiology, but essentially that's all you need to know about ground tissue for now. So these three tissues fundamentally make up plants. The leaf, the root, and the stem are all comprised of the vascular tissue running along the middles, the dermal tissue of the exterior of the thing, and the ground tissue comprising everything in between. So we'll see these three fundamental tissues during plant development and growth a lot. And because their tissue is so organized, the way that it is arranged throughout different plants is very useful for identification. It also comes up in just regular plant biology problems sometimes, so I thought I would touch on it a little bit, specifically steles. When plants have specific vascular tissue structures within their stem or roots, we can analyze how it's arranged in a cross-section, which is called a stele. Early vascular plants had a thing called a protostele, but now most have a siphonostele. A protostele is basically the cross-section where there is a single core of vascular tissue. This gray is vascular tissue running throughout the entire length of the stalk. Siphonosteles and eusteles have more of a central core of ground tissue instead, and the vascular tissue is instead delegated to kind of a ring cylinder, kind of hollow cylinder of the stalk, which of course helps with surface area, but is also just a byproduct of how the more derived vascular plants evolved microphylls, which are a type of leaf that I will talk about shortly. The Lycophytes, or clubmosses, and their relatives typically exhibit a protostele, which a more specifically a plectostele, but I don't know how important that is to know. And ferns exhibit the siphonostele, or dictyostele, or solenostele. This is kind of a map of how these steles evolved from the archetypal protostele in ancient ancestors of vascular plants. And therefore, steles are actually very useful not only for ID, but also for elucidating these kind of evolutionary relationships. For example, a long time ago, plant biologists thought that vascular plants and their eusteles evolved directly from the siphonosteles of ferns. But actually, through examining the ancestors of ferns, vascular plants, and seed plants, they were able to figure out that the eustele of seed plants was derived directly from the ancient protostele and did not emerge out of the fern, which means that seed plants also did not emerge out of the fern, but instead diverged early from the original ancestor of vascular plants, which is really cool. And paleobotany, that field that studies fossilized plants, is able to use this kind of architecture in ancient plants, which can be preserved very well if petrified, to figure out that kind of stuff. So that's just something that I thought was cool. Oh, also, another quick point involving the siphonostele and eustele is that because they have ground tissue both interior and exterior to the vascular tissue ring, they are given different names. So the ground tissue in the interior is called the pith, and the ground tissue to the exterior is called the cortex. So ferns, also known as Monilophytes or polypodiophytes, and clubmosses and their relatives, which are known as Lycophytes, are frequently grouped together as Pteridophytes, cryptogams, or the seedless vascular plants. Thank you, plant taxonomists, for your naming schemes. Without seeds, they must use spores to reproduce, and they also still have flagellated sperm, which you might remember as an ancestral trait from when plants were algae, and therefore require a film of moisture to reproduce, so they still require moisture. They can stand up very tall now, thanks to their vascular tissue. This is Lepidodendron, which is an ancient lycophyte in the Carboniferous. It was the main component of those big forests, and it is a great example of a big woody lycophyte, even though its wood might not be true wood in a sense. I don't know, that's not important. Another ancient ancestor of vascular plants is Rhynia, which along with the Lepidodendron helps exhibit this kind of dichotomous branching within really ancient vascular plants. So instead of lateral branching in modern plants where there's kind of a main axis and branches just come off of it, smaller branches come off of it, the main axis splits into two symmetrical axes, forming this kind of fork. And it's ugly. I really don't like symmetry within my plant. Lycophytes diverged earlier within the Pteridophytes, at least, and retain ancestral traits, notably microphylls, which are a type of kind of archaic leaf. They also have the protostele, which is a little bit more ancient than the siphonostele of ferns. We also start to see a lot of spore innovation through heterospory and endospory. Heterospory is when two different types of spores are produced that differentiate into male and female gametophytes, respectively. They also exhibit they are polysporangiate in that multiple sporangia can be born on one individual sporophyte. You might remember that in bryophytes, each sporophyte only had one capsule or one sporangium, but now they can have many, whether clustered on leaves or in strobili, which are these cones. So this kind of spore innovation, oh, and I didn't talk about endospory. Endospory is also seen in some specific pteridophytes, and it is where the gametophyte is retained entirely within the spore. It is usually seen in heterosporous plants. But anyways, all this kind of tinkering with spores is kind of a clue that the plants were not exactly happy with spores. Spores are not exactly the most efficient mode of reproduction. So just keep that in mind as we continue discussing evolution towards seed plants. So sporophylls and megaphylls, I will touch on briefly. Sporophylls are modified leaves that bear sporangia. Microphylls and megaphylls are two distinct types of leaves, okay? Microphylls are found in those Lycophytes, and they're more ancestral. They are small, which is why they're called microphylls, but importantly, they only have one single strand of vascular tissue, and therefore are usually associated with protosteles. Megaphylls, on the other hand, are much larger and associated with the ferns and the seed plants, which have siphonosteles and eusteles. Siphonosteles and eusteles have gaps called leaf gaps where megaphylls arise. And also megaphylls, the leaves have a branching vein system, which means that the vein, there's not one single strand of vein of the leaf, it branches. Megaphylls define this clade called the euphylophytes, which includes ferns and seed plants, but not lycophytes. Okay, so here is an example of a microphyll and how it could have arisen as an offshoot of the protostele. And it is theorized that megaphylls kind of arised out of the combination of branching stems. And when a leaf forms around it, it creates that branching sort of vascular structure. Here are sporophylls. I'm not really sure why they're down here, but sporophylls are these modified leaves that have sporangia on them. In the case of clubmosses and some ferns, they are wrapped into cones called strobili. So these are modified sporophylls that have sporangia and are wrapped into these kind of cones. And the euphylophytes are this kind of clade of organisms where the tracheophytes arise here. So tracheophytes can be split into two clades, the lycophytes and the euphylophytes, but they can also be split into the seed plants, the spermatophytes and the pteridophytes, but the pteridophytes are not a clade. There's a lot of ways to split this, and the systematics can be kind of confusing. So the classic pteridophytes that will most likely be talked about are clubmosses and ferns, more specifically leptosporangiate ferns. Here is a clubmoss and a traditional leptosporangiate fern. That's probably what you think of when someone says fern. Tracheophytes are sporophyte dominant, like I said, and what's unique about pteridophytes is that although they have sporophyte dominant, you can still find their gametophyte, and it is actually free living. It is a separate little plant, and it's very cute. It's called the protosteles. So here's a fern gametophyte, which is probably the best example of a free living gametophyte because you can even usually find it on the forest floor sometimes. And the little sporophyte actually grows out of the tip of it. Clubmosses and some other ferns have their gametophytes underground, which actually form mutualistic relationships with fungi for food, which is pretty cool, but of course you won't see them. As the seedless vascular plants, they use spores and flagellated sperm to reproduce, like I said, and their spores are borne on sporangia, on those strobili of the clubmosses back here, and the ferns have their sporophylls with clusters of sporangia called sori, and these sori are covered with a little membrane called the indusia. A little bit of vocab there. Some other really cool pteridophytes here are isoetes and selaginella. Selaginella are spikemosses, and they're kind of spiky, I guess. And isoetes are called quillworts, and they're aquatic. They also exhibit CAM photosynthesis, CAM photosynthesis, which they evolved independently. Another thing that these pteridophytes evolved independently is heterospory, which I talked about, and it is kind of just showing how beneficial heterospory is, that it is evolving independently in other lineages of plants. These are more related to clubmosses. So when I say clubmosses and relatives, these are the relatives. Fern relatives, on the other hand, are kind of gnarly looking. Here's ophioglossum, which is part of Ophioglossum. That family of plants are all really interesting because they usually have one leaf, just one really big leaf, which is pretty interesting. And they might even have a smaller leaf. It's not really a leaf, it's a modified leaf, the sporophylls that are clustered and create sporangia and spores. It's also called an adder's tongue. It looks a little bit like Araceae, which is a monocot family, I believe. Doesn't really look like a fern when you think of fern. Another thing that doesn't look like a fern is psilotum, which is also called a whisk fern because it kind of looks like a whisk, I suppose. It has its leaves almost completely reduced and so photosynthesizes with its stem instead, and these sporangia are kind of just in these balls of three. I don't know, it's really interesting. It also exhibits dichotomous branching, which is one of those more ancestral traits. And then there is equisetum, which are horsetails. Not really true ferns in any way, but I guess they look like horsetails. Oh, I don't know. They also have photosynthetic stems, even though their leaves are kind of present. These are vegetative shoots, which means they don't have sporangia. A lot of these ferns actually kind of switch between a vegetative leaf and a more fertile leaf, depending on whether they are sporangia or not. In general, though, these are more related to ferns, and these are some eusporangiate ferns as compared to the leptosporangiate ferns, which are more classic ferns, I suppose. Interesting about horsetails is that I see them pop up sometimes in like competitive biology problems for some reason. Horsetails contain high levels of thiaminase, which breaks down vitamin B1 and therefore causes death. Or beriberi is the appropriate syndrome. And so maybe there's a problem where a bunch of livestock are dying from eating some weird grass and it's horsetails, and it's because their vitamin B1 is getting broken down. I don't really know. Maybe you'll see that on a test someday. So heterospory is this innovation as we start moving towards seed plants. And heterospory is essentially where the sporophyte produces two distinct types of spores, the megaspore and the microspore. You can also just think of mega as meaning female or like towards the egg cell lineage, and microspore as male towards kind of the pollen or sperm cell lineage. So while the homosporous plant produces a single type of spore and a gametophyte that produces both gametes, therefore it can self-fertilize, which can be useful for these kinds of seedless vascular plants that, you know, there's no guarantee if you'll have the two sexes of gametophyte landing next to each other, if that makes sense. But in general, it's more advantageous for seed plants to be heterospores, producing two types of gametophytes to promote outcrossing and genetic diversity that will produce one type of gamete respectively, that when fertilized will create the zygote. Heterospory is seen in these Lycophytes and also a few aquatic ferns as well. And here's a quick life cycle recap of the Pteridophytes. It's very similar. Well, I don't know about very similar, but generally the principle is here, and we get to see some alternation of generation in action. So the sporophyte, when mature, will have sporangia bore on its sporophytes, the microphylls, reproductive leaves. They will release spores. In this case, it's a fern, so it's homosporous, producing one type of spore that will germinate into the gametophyte, the bisexual gametophyte in this case, because it produces both sperm and archegonia. Sperm and egg. Sperm in the antheridia and egg in the archegonia. That sperm has to swim through some moisture to get to the egg. It has flagella, but when it does, it will fertilize the egg into a zygote, and that embryo is carried inside of the gametophyte. Remember, these are all embryophytes, so the embryo is retained within the gametophyte, which is very nice of the gametophyte to do, and the cycle starts itself over again. Very wonderful. So a quick question here. We're going to continue LARPing as plants. If you were a discontent pteridophyte, what would you complain about most? Would it be that your free-living gametophytes are much too vulnerable to the environment? Would you say that your spores are inefficient because they don't disperse very far and give your offspring barely any nutrients to survive? Maybe your sperm will require moisture to swim to the egg, which is just plain annoying. For fertilization to occur, your gametes have to be produced at the same time and place, temporal and spatial. Or maybe she's not responding to your texts. Well, what do you think? The correct answer here is F. A, B, C, and D. Spores are just not very good, okay? Not only do they have to germinate into gametophytes by themselves far away, they don't even get very far away, and there's no guarantee that they will germinate into a gametophyte because spores don't really give the gametophyte anything to work with. They just land and hope that it works. And so for that reason, they're very inefficient. Of course, then the gametophytes, if they do live and produce those gametes, not only do they require moisture so that the sperm can swim to the egg, but that there is a stipulation that gametes have to be produced at the same time and place. There's no guarantee that your gametophyte will even be able to fertilize anything if there is no egg being produced for the sperm to swim to, right? Or maybe the egg is not produced at the same time as the sperm and you have this whole kind of big issue. It's just that these spores just suck, and I really wish that there was a way to kind of improve on this design. I just don't know what to do. Wait, what's that sound?
There's a star